Um, okay. So it's day 4. You're all experts in MD. We get a lot of questions about now that I can run MD and I can run a lot of MD, and you heard about the MD3 and evolution of hardware and software. The question that starts coming up is, what do you do with this pile of trajectories that you now have, right? How do you extract information from it? And that's what I'm going to try to focus on for a little while, and then we can talk more over the hands-on session later today. Um, so you've learned how to run and prepare and do some analysis on that will cover things like the dynamics of protein structures and molecular structures, in some cases charge with distribution and polarization, lots of techniques to measure changes in free energy. And yeah, the question becomes descriptors. What are the descriptors? What do we actually measure? There's some very intuitive things that you can, well, you know, if you use QuickMD, some of these descriptors are going to be buttons, very easy. You click on it and you get, you know, RMSDs or some geometric measurements. Others you may have to work a little bit, but it's still pretty straightforward to measure. They're ready-made tools that will give you, for example, volumes of channels over a transmembrane channel, for example, like you heard about before. But then once you measure these things, you start wanting to ask interesting questions. So nowadays, it's very easy to see in drug development people trying to use these measures to try to create relationships between changes in a molecular structure, or changes in the interaction between two molecules, and try to extrapolate some activity measurements for drug development, for example. And of course, the AI models, all the big and famous ones, they rely a lot on IMSDs, very straightforward comparisons of structures, right? With that said, you want to do cool science, you want to do new science, that means you want to start asking new questions. That usually means there is no software that you can just click a button on and it will give you a new question, right? Or a new answer. So um, that's something that I focus on a lot. I really like asking new questions. That usually means writing new code, and that usually means understanding the biochemistry and trying to put that together with the data that you already have. And yeah, and so this is one major question. How can we just create new representations to describe molecular interactions? So yeah, this stuff you're all experts in already. But then let's go back and think about what's happening in a molecular dynamics simulation. Where you have your data, you have your DCD from NAMD. It's a lot of XYZ positions for a lot of atoms, right? It's pretty straightforward, simple data. These atoms are going to be moving around, vibrating, but we know that these vibrations are not isolated, not random and dissociated from one another. Atoms will interact with one another, either directly, where they're bonded to one another, so there's a vibrational frequency because they're bond, or because they're part of a larger molecule, and even though they're not directly connected, they're participating in rigid motions, or they are interacting between a ligand and a protein, and they're going to be non-bonded forces that also affect how they interact. So their movement is correlated, right? Their movement is not completely dissociated. There's stochasticity, but it's biased. So how do you get to that? And there are many different ways to look at and try to extract correlations from molecular dynamic simulations. One path, one direction, class of methods focuses on calculating correlations between interacting atoms. And one of these is called dynamical network analysis. And this is, it's really a methodology that was developed about 15, 17 years ago now. And the basic idea is you have these XYZ positions. Over time, you can look at two atoms and you can calculate how correlated is the motion, right, between those two, the two atoms. And from this correlation, we can start inferring information transfer. We can start inferring how they are influencing one another. And this kind of technique has been, well, it was originally applied to find communication pathways between a regulatory site on one side of an enzyme and a catalytic site on the other side of an enzyme, finding how these two distant areas of a protein communicate with one another. Yes.
So there are autocorrelation functions where
Yeah.
you can do individual, atom correlation.
Sure, autocorrelation. Sure,
right.
Yeah.
I mean, so what is is it the network analysis is distinctly different, and in what ways is it?
We'll talk more about, yeah, okay. We'll go more into detail. And yeah, just the original idea, the point here is not to focus on autocorrelation, but on correlation between motions of different molecules, or at least different residues. So one application is finding what are the steps, what are the individual interactions between distant areas of a complex. Another one is to look at interfaces between subunits of a macromolecular complex, for example. That's what I like to do a lot. I have fun doing that. And so the way you do that is this cartoonish representation here of a couple alpha helices. Let me bring my mouse. Yeah, it's a couple turns of an alpha helix and a beta strand here. What dynamical network analysis does is it represents every amino acid as a group of atoms that gets represented by a single atom. In amino acids, that's usually the alpha carbon. And you can track all the atoms of this residue and see which other residues are within close proximity or directly interacting with that residue. And because you have a molecular dynamics simulation, because you have the positions of all these atoms over time, you can calculate the correlation between the alpha carbons of these nearby residues with the central residue you're looking at. And you can then transform and calculate these correlations between pairs of nearby residues. Do that for every single residue of your protein and transform your protein, complex, ligands, everything into a network, into a graph, where the edges have weights that are the correlations between the motions of those residues. Right? So you're really transforming, creating a new representation for all those, 500,000 atoms moving around in space to a graph, to a network with edges. Yeah.
You mean the edges means, is it like a distance between like 3 alpha?
So it isn't a distance. It's really, you have the position of one C alpha over time, you have the position of the other C alpha over time, and they're moving around. And then you can calculate how correlated is the motion between those two C alphas. So if these are two alanines, right, one in one beta strand and one in another, they won't have a really high interaction. So there will be a smaller correlation between their motion. But if they're opposite charged, if they form a hydrogen bond, or if they're backbone hydrogen bonds, then the correlations, well, there will be a high correlation between their motions. And then you can infer that they are interacting more strongly. There's a biological significance to that interaction. Does that make sense?
It's possible
the
unexpected correlation could be found by this way. That's the unexpected non-hydrogen bonding.
That's the best case scenario. Right? Best case scenario. That it shows you something that is not obvious. That is that just by eyeball you can't really notice, but yeah. And we can do that. The obvious cases, amino acids. We usually represent those through their alpha carbons. If you have so nucleotides, for example, are larger, you can split larger residues into smaller groups of atoms and represent them. For example, one by the nitrogen for the nitrogenous base and the other by the phosphate. If you have a large ligand or some drug or a cofactor, you can split that in smaller groups and study individual subregions of a large cofactor. That's all there. And you can then scale this up and look at interactions between subgroups of a big molecule. For example, you may have this is the mitochondrial complex 4. It's multiple subunits embedded in a membrane. You may be interested in interactions between multiple of these subunits, right? And we can definitely look at all of them individually and see what their interfaces look like. But you can also look at them and look at the embedded membrane and identify lipids that have, strong interactions, high correlations with amino acids in those subunits. So you can start identifying lipids that are structurally relevant or physiologically relevant. So yeah, we've been using this for a little while. Rafael mentioned a couple of examples. One recent one, which unfortunately is very familiar to everyone. I don't need to really explain what COVID is at this point, but we've heard a lot about the interactions between the receptor binding domain, the tip of the spike protein of COVID, where it interacts with ACE2, the human molecular target, right? So one thing we can do, and then one thing that our collaborators did, was to isolate the receptor binding domain and the ACE2 and tie those two subunits together and attach them to a magnetic tweezer experiment, and basically bring them together and pull them apart over and over again, and also hold them under constant force, different constant forces. And what this experiment shows you, and this is experimental data, right? What the experiment shows you is if you apply a little bit of force on this pair of proteins, with a little bit of force, the proteins stay bound together for most of the time. But then you can increase the magnetic force, you can increase the force applied on this pair of proteins, and then what happens is, well, they start dissociating for longer. And you can see here in this trace, the distance between the two proteins. It increases for a little while, but then they bind again and they stay bound for longer. But as you increase the force, you can see that you bias this dynamics to an unbinded state, right? The unbound states. And this happens over what? A couple piconewtons? Very sensitive. So this represents how resilient that molecular interface is to external forces, and you can measure that directly. So how do you track that with computational work? What can a molecular dynamics simulation give you to help you understand this process, right? We can simulate them, and we can simulate them comparing SARS-CoV-2 and SARS-CoV-1. So experimentally, when you compare SARS-CoV-2 and CoV-1, one interesting thing that happens is you know that CoV-1 here is in blue and CoV-2 is in green. So the first thing you know is that under the same forces, SARS-CoV-2 evolved to be more mechanoresilient. That means that under the same forces, CoV-2 stays bound to its molecular, to ACE2, to the molecular target for longer. Something happened during evolution that made it more resilient to external forces. So what can we do? We can look at the same system where we're not doing magnetic tweezers, but we're doing MD simulations with exactly the same system, applying this external force. And we can start looking at the correlations between the motions of residues on ACE2 and on the spike protein, on the RBD. With this, we're comparing the correlations between the two strains, or the old one and the current one. We can see that some mutations that happen along the way really affect how correlated the motion is between residues across that interface. So somehow over evolution, the RBD changed and the mutations caused that interface to become more stable. It didn't happen by increasing the correlation all over the interface, which was part of the unexpected. Naively, I thought, well, if COV2 is more stable, then we're going to see an increase in correlation all over the surface. Didn't happen. That was an interesting, unexpected event. Right? A couple of mutations actually decreased correlation. But overall, the interface became more stable. So counterintuitive. And then looking at these mutations and thinking, well, what happened over, I forgot how many years between COV1 and 2, 10, 12 years, or what happened, almost 20? What happened there was, well, there weren't just mutations on the surface. There were mutations all over the receptor binding domain and all over the virus, right? So the question becomes, what is really happening? We're looking at the interface, that's interesting, but maybe the mutations that happen inside the receptor binding domain had a bigger impact in the stability, the mechanostability of that interface than the ones on this interface. So Priscilla looked at this and thought, okay, let's check, right? We have the interface, we can see which ones are more correlated or not. And we know exactly what the amino acids are and where they are. So let's mutate SARS-CoV-1 only along the surface so that it looks like SARS-CoV-2, but retains all the internal mutations of that retains all the original sequence for SARS-CoV-1. And our collaborators synthesized this chimera and tested the mechanical resilience. And what happened that was really interesting was, well, you know, COV1 was weaker than COV2, but this chimera that only had the surface looking like COV1 actually became a lot stronger, almost as strong as SARS-CoV-2. So this really shows that the interface bears most of the relevance for that mechanostability. The core of the protein was still very different, but so that was an interesting, unexpected set of observations that came out of running a lot of MD simulations, measuring, creating this extra measure, focusing on the interface and identifying points where you can, like Raphael said, test, right? make a hypothesis from the simulations and then go back to the bench and see what happens. Still more or less on the same thing, thinking about COVID, the next thing we did was to look at SARS-CoV-2 variants and how they quickly evolved over just a couple of years. And so one thing we can do is thinking about the same setup, right? We're just using the molecular dynamic simulation of the receptor binding domain of the spike protein, the ACE2, but now we can change to track the variants, and we can look at the interface and see which residues that got mutated had their correlations change significantly. And again, it isn't obvious because sometimes one residue changes, mutates, but it affects the surrounding residues. So maybe you don't have a mutation somewhere, but that amino acid becomes a lot more important or a lot less important for an interaction. And you can track that because you're tracking everything along the surface, right? It's MD, you know every single atom all the time. So we can definitely talk about the individual correlations. We can look at all the correlations between all contacts in the ACE2, between the ACE2 and the RBD. And we can add them all together to calculate something we call a total correlation, which is the sum of all of those individual correlations across the interface. If we normalize everything by the wild type, make that the total correlation 1, we can compare the other variants of concern. And at least computationally, we saw that the alpha variant had a significantly higher correlation across the interface. And interestingly, our collaborators could measure the mechanical resilience of all these variants of concern, and they saw a very, very similar pattern. They also saw the alpha being significantly more resilient than the other variants. Putting this in context with evolution and virology and how that affects infection is the next steps, right? But we can definitely go from single atoms to more global effects by doing that. And like I mentioned before, we can look at the interface, we can look at the surface of the receptor binding domain and think about how the different mutations affect that interface. A lot of these mutations that happen in the variants of concern didn't happen at the interface. But if we have a way to represent the overall interface, and this is just a 2D representation. It's a PCA transform map of this interface, which, as you know, the protein isn't flat, right? There's some topology there. But imagine you could just project that surface on a flat 2D representation. It will look something like this. And you can definitely calculate the correlations between the residues along that interface, right? And if you do that and compare with other variants, you can see how mutations affect the interactions, how cohesive, how cooperative are the interactions on the surface of the SARS-CoV-2 RBD itself. Right? So there are ways to measure the effect that may, again, just by intuitively looking at an MD simulation, we would never be able to capture. But it definitely tracks with mutations and with effects that we know we can measure. So going to another example, in this one, the one that Raphael mentioned, the SDRG, the adesin, and how it has these different paths of unbinding. Like he mentioned, he talked about how experiments can be done with relatively low force, and you can just apply a constant tension across the complex, or how you can apply a really large force to unbind it, and how these two are different paths. One thing we can do is measure how this force propagates over the complex. And when we run the simulations, we can definitely run simulations under constant low force, replicating a magnetic tweezer experiment. And we can run simulations with a very strong increasing force, replicating an atomic force microscopy experiment. As we can run simulations on these two kinds of creating these two kinds of contexts for the protein. And then we can calculate correlations across residues for the entire system and see how the force propagates over the entire system. And not surprisingly, it's different because we know that the dissociation is different. That's not very surprising. But what's interesting is because using this kind of methodology, you can see exactly which residues are involved in these different dissociation pathways. So this, you've seen this, that you have to run hundreds or even thousands of simulations if you're trying to connect large-scale behaviors over a large span of forces. There's an inherent stochasticity there that requires sampling so that we can properly assess what's happening at every stage. The catch is you need a lot of computer power to do that, or you need a lot of time, and you need to produce all of this results. And then you move to a different problem, which is the kind of problem that I like, which is more of the data science kind, right? You have to aggregate and prepare all of these data. It doesn't fit on a spreadsheet anymore. Right? You have to do a little bit of organization, a little bit of codes to organize these piles of data in a way that you can use. Then it's interesting, it's important to have methods to do interactive data analysis, because there's some exploration there that, again, it isn't obvious. You're looking at the whole system. A priori, you don't know what's important and what's not. So there have to be methods to allow you to interactively analyze data. That's where VMD helps you a lot, and that's where other techniques help you a lot. And ultimately, you can combine these data into training data for machine learning models to help you make clustering or unsupervised learning, or to actually do predictions that match, for example, experimental observations. And then we can circle back to QSAR, or we can circle back to structure activity relationships, right? And other interesting things. So in this case, Rafael put this plot on his slide. It took me a while to figure out what to plot. Right? This wasn't obvious. So, I mean, we know that this complex has a peptide that gets buried under two different domains with another beta strand that goes on top of it. There's a lot going on in there. It isn't obvious which region, which interactions, what are you actually going to look for? So sure, this one we knew that there were a series of hydrogen bonds. That's a good place to start, right? So you have to build that over time, but there's another region of this protein that's over here. That is where, that's called the latch, which is where the extension of the first module that I'm painting in green here, it extends over the human peptide in orange. Yeah, the adhesin, the bacteria adhesin binds to, right? It extends over the human peptide target, and then it binds on the other bacterial adhesin subunits. So this region here seemed to be pretty important, but how important, right? And then you can do some exploration, and you can see that under force, comparing the correlations between the residues of the latch and the subunits, the same thing happens as the correlations between the human peptide targets and the surrounding bacterial adhesin. The correlations increase with force. It wasn't obvious at all, but it happens, and it happens very, very clearly. There are a couple of red spots here, which are residues that where the correlation decreased. Those are interactions that get lost when you increase force, but the vast majority of the interactions increase significantly. And that was super interesting. And then, yeah, okay, so now that we know that these two areas are important, maybe we can test this hypothesis. How can I test this hypothesis? Well, one thing I can do is I can get these data, only the correlation data, right? It's a number between 0 and 1. It's a correlation of motion between residues in the human peptide or the latch and the bacterial adhesins. Like only using that, no physical chemical data at all. You can create training data and train a few different machine learning models to predict something that we know, which is the rupture force, right? Because we're pulling these proteins apart. They do detach at some point, and you know the force that you use to detach them. So you can do that at different velocities like we did here. They would detach over a huge range of forces, and then you can try to predict, can I predict what the force is if I know what the interaction, what the correlations are? Turns out you can't. So these correlations do contain information that allow you to extract mechanical stability data, right? Without knowing what the amino acids are, without knowing geometry, without knowing anything, just looking at the correlations. So there's a lot of information in there. That made me happy. So, okay, let's try it again. Let's try on another system, a system we thought was a lot simpler than this complicated knot thing that goes over the other. Let's look at a very simple system, which is one that we know well. It's a cohesing Dockering pair from cellulosomes. Things have been studied for decades. This is not complicated at all. It's a couple of alpha helices on one side and a beta sheet on the other. It looks pretty straightforward. Again, we can run a couple hundred simulations. And if you build the simulations, it's a small system, you build the simulations, you prepare them exactly in the same way, and you start them independently, and over 200 simulations, you get this huge range of possible rupture forces, or forces that you need to apply to pull the proteins apart. Even with everything really well controlled, starting from the same point, stochasticity is there, right? It gives you a huge range of results. So one thing we can, now that I know that I can use correlations to predict mechanical stability, how strong, how much force you need to apply to dissociate these proteins, how soon can I do that, right? How much MD simulation do I need to run to make this sort of prediction? Because I'm cheap. I don't want to run long simulations. I want very short simulations, as little as possible. And one thing we can do is we can align all of those traces that I was showing you before. We can basically align all of them to the rupture point, to their maximum forces. And then we can look at what was happening with the system at different windows, right? With these blue rectangles here are simulation windows. We can get a window right before the rupture point, or we can get a window a little before that, or a little before that, and we can do that on 11 different windows, all the way from, very small forces. And then we can go back and train do the same idea, right? Run network analysis, capture the correlations across that interface, transform that into training data, and train a dozen different machine learning models and see what's the best prediction you can get. And turns out, even at very early windows in the simulations, you already have a very good correlation. This is Spearman rank coefficients, rank order coefficients. It tells you how well you can rank the strongest to the weakest of the simulations. And you can do that really consistently very early on in the simulations. So even with relatively small forces, you are already, and it gets back to your question, even with small forces, you're already biasing the trajectory, right? The behavior of the complex to a stronger or a weaker direction, right? So even with small forces, and that gets captured by correlations of motion. So essentially, I didn't have to run all the way here. I could have run half the simulations, right? But we didn't know that. Now we know that. And at least for this system, it works really consistently. What this plot doesn't show you is that there's quite a bit of exploration, quite a bit of code before that has to happen before. You need to prepare 200 simulations systematically. You need to load all that data, extract different portions of those trajectories, transform and analyze all of them using dynamical network analysis, and then train models on top of them. There were over 1,000 stretches of simulation that had to be analyzed for this plot, that simple plot to happen. It isn't complicated. It's just a loop. It's a for loop, right? It's from 0 to 200, you do something. But there's a little bit of exploration that has to happen. So we start with QuickMD, we build a simulation, explore with a couple of simple descriptors, get an idea of what to do, and then move on to the more complex things as you explore, as the data grows, as the questions grow. And yeah, so this is exactly half an hour. I'm going to stop here. I won't show you